Next up is Ben Alexander from the Broad. Ben is a principal software developer and has been working in that arena for some time.
I'm going to present to you today. This talk was originally going to be presented by my boss, Steve Bruds from the Broad Institute, but I'm going to take a slide deck and see if I can do it justice. I come from the Broad Institute. We have been using ChemAxon for a long time. We're enthusiastic users, and it is at the core of our informatics base, what we use to handle a large quantity of data, which we generate and then use to perform lead optimization and all the rest. So let me start by providing a few words. So here's the Broad Institute. The Broad Institute was developed, started about 7 or 8 years ago, and it was built as a conglomeration between several different institutions. Harvard, MIT, and then some area hospitals got together with a generous donation from Eli and Edith Broad, who gave us, over the course of a couple of installations, $600 million, and allowed us to start this gigantic endeavor with the goal of trying to investigate intractable biological problems and come to some sort of solution. The way that the Broad approaches these challenges is twofold. We have a series of programs, and the idea of the programs is that they are attempts by multiple scientists to work together. The scientists might be academically funded, and they are then able to address problems such as cancer, chemical biology, infectious diseases, and we can address problems thereby that are outside of the purview of what commercial pharma would generally attack. So, for example, there's a large malaria program. In addition to the programs, there are platforms, and the platforms provide services that can support either the programs directly, or they also can work in an outward-facing way to provide services to the larger community. So, for example, we have a large room full of DNA analyzers, and we are now reading something like 4 terabases a day of genetic sequences and storing lots of information thereby, along with lots and lots of screening. Now, the part of this that I'm involved with is the chemical biology platform and novel therapeutics, and so that's what I'm going to be talking about in my presentation here today. Why do we have a Broad? The Broad is here because so many of the problems that need to be addressed are difficult to address and require large numbers of resources. There are a huge number of targets that are classically considered undruggable. Either these are proteins that don't have that small cavity that we would hope a small molecule could interact with, or else they are immune to biologics because they are inside the cell and are kept out by the cell membrane. Or perhaps we're talking about complex biology that we don't understand very well and we need empirical information in order to really address. Either way, or in many other ways, sometimes it requires the combined resources of a large number of scientists and equipment, and ideally, or in our case, in a nonprofit environment in order to address some of these problems. So the platform that I work on is our chemical biology platform. It has been around not for all that long. It started with some early DOS concepts, and this is diversity-oriented synthesis, not a reference to an antiquated operating system from the late 80s. As well, we received an NCI initiative. The Broad started in about 2004, and then we came up with our first library. So we're doing CombiChem to generate diverse sets of compounds, and then we organized all the robots to start screening all of those, and then the Broad broke away from MIT, which was providing its initial source of funding and facilities, and became its own nonprofit organization as of 2009. And as of today, we have 150,000 of our own compounds that we've developed, and then we have a number of other compounds that we get through the MLPCN program, which I'll talk about in just a minute. So screening is something that we spend a lot of time doing. So we have a large room full of robots that go day and night taking well plates in a variety of formats and running different kinds of assays. When we're lucky, these assays produce results that can be quantified in ways such as in the plot that you see here, where maybe natural products go shunting off to one side and different other collections of chemicals show up in different ways. But all of this screening generates a vast amount of data, especially when we want to take those initial screens and use them to then organize secondary screens where we perform at-dose work and then allow maybe a third set, tertiary screens that use slower assays. All of these put together requires a great deal of data in, data out, and data analysis. And that's where, of course, ChemAxon comes in. One more program to describe before I proceed is the MLPCN program. This is something coming out of NIH. And they give us 324,000 compounds and require that we develop assays or use other recognized assays. And we only get paid by them if we run the assay on at least 300,000 compounds. So we need to run through our assays, and then the goal is to produce either publications or to generate probes. Everything that we generate after an initial waiting period of 6 months goes straight into the public realm through PubChem. So our first shot at trying to take this data and do something with it was CBIP version 1, which we had running as of about a year and a half ago, after we finished our transition from our previous Informatics tool maker, which was Daylight. We switched over to ChemAxon, and now we are much happier. So here is, let me run through this quickly for you. So first you pick out a set of reagents. You then need to design a library. And if you can do that in some sort of a virtual way before you actually build the library, that's advantageous. And some aspects of the Marcouche enumeration, as we talked about during yesterday's session, are useful here. Though we also need to use Reactor to do a step-by-step representation of the syntheses. Then we want to use our ELN in order to describe the chemistry in more detail that we're going to undertake. Then we go ahead and undertake the production of the library. We're using, most of the time we've been using solid phase synthesis with RFID tags to tag the compounds as they produced. Now as they are produced, now more and more we are going to solution phase chemistry as well. Then we need to take the compounds that we produced, we cleave them in the case of the solid phase synthesis, and send them off to our analytical shop. If we can achieve at least 70% purity, then we move on to the next stage of generating various analytical numbers that we can then present to the scientists. And if all that goes as planned, each of those compounds goes into compound registration, which I've heard a lot about this morning. We have our own set of tools for compound registration. Then we store them in big freezers and allow people to access them through our compound management tools. And then we set up our screens, and the screens involve a big room full of robots, which then take the compounds and run through some large number of them, generating screening data, which our scientists then analyze. And then then they take usually some subset of the hits and then either expand them through similarity searches or decrease them through cherry picking, and then go back in and perform the secondary screens. I'll talk about that in a little bit more detail later on. So what's the basic architecture of the software that we use to do this? Here is CBIP1 as of about 18 months ago. Here the blue squares represent software that we've written, and the yellow squares represent software that's third party. And so this is not organized. The previous slide was workflow. This is organized more. I think the columns have some meaning here, but more. I think the main lesson to draw from this slide is that we have a bunch of different components, many of which use ChemAxon in one way or another. Here's the chemical cartridge, and all of these need to integrate. And this is the great challenge of what we do, is to provide our scientists with capable, highly friendly and interactive tools, and yet to be able to integrate all of that information into a larger system. And then up here we have the tools that they can use to analyze the data that they've generated elsewhere. Here now, and here's a list of the ChemAxon tools that we're using, or were using as of 18 months ago. The list only increases. Here is our newer version, which is pretty close to the same. Let me see if I can do this. You can see they're pretty close to the same. I can't do that very well, but we have a new tool here, which is our SSAR visualization tool. This is stereo-specific structure-activity relationships, and I'll show that tool in a little bit more detail. We have made our own compound ordering system. We have a way to publish the data to PubChem, and we've now gone to the TIPCO version of Spotfire, which has some increased capabilities over our original use of Spotfire. And again, note that ChemAxon plays a critical role throughout our infrastructure of computing. Okay, so about this stereo-specific SAR, this has particular relevance at the Broad because stereo-specific chemistry is important to us. Following the work of Dr. Stuart Schreiber, we have engaged in syntheses that are stereo-specific. And one of the tools that we use to achieve that is a build-couple-pair technology. The approach for CombiChem, more typically, as I'm sure many of you know, is to take a core, a complicated core potentially, with a few attachment sites, and then to put on a large number of fairly simple substituents at those attachment points. This strategy instead involves building up more complicated building blocks that may be stereo-specific, that are stereo-specific. We then couple them together, and so now we have a series of different building blocks, even more complicated, again, all stereo-specific. And then in a final pairing step, we bring in another chemical and then perform this final step. I am not, by training, a synthetic chemist, but to my eye, it's clear that what's always going on here is a intramolecular cyclization step. I don't know if that's absolutely required for build couple pair or not, but you can see that what we have are a large number of stereo-specific molecules. And in fact, if, for example, in a molecule we have three chiral centers, we will endeavor to make all 8 of the possible stereoisomers and then test them all. And so having all of these different stereoisomers, how do you look at them? What's a good tool for visualization? Well, here's our architecture from before, and I want to talk a little bit about this tool. So here is our SSAR visualization tool, and what's going on here is, imagine that you have a core that's represented here. You have a list of substituents, so we're talking about 2 attachment points on this core, and then you have, for each one of these blocks, you have 8 different areas, each of which describes a different organization of stereochemical combinations. So maybe one of these is RSS, SRR, etc. And you will find instances where you get pronounced activity in one of those, or one or more of those arrangements, and not at all in other cases. Now, in not all cases are all combinations possible. Certainly there are cases where things just don't fit together. If you're trying to have the bonds pointing out of the carbons in particular ways. And there are other times when you have enantiomeric pairs where you're going to get the same results, but often you do get diastereomers where the results are quite different. And so this is one of the areas where the Broad, I think, is doing very interesting work. Having developed this visualization tool, you can also use it for QC data, as is shown in this slide. Let's talk about another part of the architecture in a little bit more detail. This time, compound registration, which is maybe apropos given the other presentations I've seen this morning. So here are some of the steps that you might undertake in a compound registration system. After getting your molecules, maybe in the form of an SD file or mole files, you then need to convert them to smiles. You need to standardize. You need to go through a standardization step, desalting, calculate some chemical properties. We perform a canonical tautomerization, and then find any molecules that have an unspecified chirality, and then we go ahead and register them. And all of this is done with ChemAxon tools, though in this picture there is one step that's done with Pipeline Pilot, just for historical reasons. But that also will change. Let's talk now about one more of the systems that we've developed, and this is TIPCO Spotfire. So this is Spotfire, but once again, it's ChemAxon in the background. So it's ChemAxon that's doing all the interesting work. So here we have a display where ChemAxon is engaged in hit calling. We can go through and pull out any groups that are undesirable. I think here we're pulling out nitroso substructures. We can filter those compounds by properties of whatever sort. Here we have a plot of mass against log P, so we're looking at Lipinski sort of things. Finally, we can find other compounds of interest, so we're performing similarity searches here. And then we finish up, and we've done our cherry pick, and we've either reduced or expanded the set of compounds that we're using coming out of a particular step, say a primary screen. So that's a list of what our system does now. Where do we want to go in the future? Well, here's a picture of where we'd like to go with version 2+. Much of this looks very similar, which is a good thing. It means that our architecture, which is used by many, is solid and is providing for our users' needs. We do want to include a data dictionary, so we're adding the various aspects of a data warehouse. Here we have another representation of that multidimensional data warehouse with hopefully different ways of getting to the data that should expand users' current capabilities. We want to rewrite that SSAR tool, since the early version, in fact, only allowed for 2 attachments, which is limiting. And we're also using Syrah by Schrodinger, and that's part of our plan to go forward. Still central is the cartridge and ChemAxon, both on the viewing end and on the production end. So what can we say in conclusion about this system, our whole informatics platform? Well, we have a bunch of thoroughly satisfied users, and the reason, in large part, we can attribute to ChemAxon. It's great to have a toolkit. It allows us to do the sort of work that we want to do, and to be able to keep that in the background when it's desirable. If we want people to be able to build their own structures, then we can surface Marvin or anything else. Our infrastructure, our programmatic infrastructure, is Java-based in large part. And that allows us to perform the integrations that I mentioned earlier, which are so integral to, or so essential to the efforts that we're trying to undertake. We find that ChemAxon's support has been excellent. They respond very quickly when there's a question or an issue. We've been able to get responses from them right away. We love the forums. Questions get resolved very quickly. And they often release new versions in order to correct any problems we may have encountered. On the flip side of that, sometimes it's tough to accommodate all of those releases, especially for the informatics substructure. That is our IT department, our database administrators. It's a little harder to get them going and upgrade to a new version of the cartridge. So sometimes we linger back a little bit, but overall, I would much rather have new and improved versions than to not have those available to us. One point, though, that has come up, and so much of this has been a plug for how much we love ChemAxon, and we do, but I've also come to make a plea, and that is, I started a project recently where we were trying to improve our ability to generate new libraries, and what we wanted to use is a new dynamic language called Groovy. Groovy runs in a JVM, so it's very much in keeping with our existing programmatic structures. You can write Groovy within Java, you can write Java within Groovy, you can call them all back and forth, it all works very well. And sitting on top of this is a system called Grails. And I don't know if anybody's, or how many people out there have used Grails, but we've had great luck with that too. Grails allows us to generate websites really quickly and move very fast. And being built upon Groovy, it allows us to access all of those dynamic programming capabilities that we're anxious to get into. Now, one of the great strengths of Grails is its plugin infrastructure, which allows people who are putting together new elements, things that they want their Grails programmers to be able to access, into plugins. And those plugins are then easily incorporated into the program you're trying to write. What we would love to get would be a plugin from ChemAxon so that somebody could say, I'm going to write a new program, a new Grails program, and throw in molecular structures and store those reactions immediately and otherwise make the data structures and the data types that are integral to ChemAxon allow programmers to treat those as if they were native data types. So that's the plea on top of all the promo. Here's a smiling picture about everybody who's been involved in this. And here is, there's Steve, the manager, who would have given this talk had he not been temporarily felled by a malevolent microorganism. Here, and there's me, but then here's all of the team. We have a split between software engineers and application engineers who are more on the science side. And here are all our happy users. That's it.